Hi all, welcome back. Myself Anjita Aravind. Today I am here with small three topics from the reproductive system, that is sex determination, development and aging of the reproductive system. So first we will talk about the sex determination process. Here our objective of the study is to understand the process of sex determination. For that, first of all we know that what is sex determination. It is the process of sex differentiation whether a particular individual will develop into male or female sex. So from this definition we understand that there is a differentiation process occurs in each individual while the development process. Then who is responsible for the differentiation process? The sex hormones are responsible for the determination of separate sexes in each individuals. We all know that the sex expression is governed by the chromosomes and genes. And when we consider a unisexual animals, that is either a male or female animals, the chromosomes are of two types, that is the autosomes and allosomes. And simply we can say that the autosomes are any chromosomes that is not a sex chromosomes, and the chromosomes which do not differ in morphology and number in both the males and females. And the allosomes are otherwise known as the sex chromosomes, and these chromosomes which differ in morphology and number in the male and females, and they contains genes and that will determine the sex of an individuals. Here we can see two symbols which is used to designate the male and females. Traditionally the symbol, that is the circle with an arrow, which designates the male, and the symbol, the circle with a cross, it is designate the females. In the human body cells, the cells which contain the nucleus, and the nucleus have 46 chromosomes arranged in 23 pairs, and both the male and females, they contain equal amount of the chromosome, that is the 23 pair of chromosomes are present in both the males and females. Out of these 23 pairs, the 22 pair are the autosomes and one pair is the sex chromosome or the allosomes. And in females, when we consider the females, they have a perfect pair of the sex chromosome, that is XX. And when we consider the male, they have a mismatches pair of the sex chromosome, that is XY. Only we have half of the DNA of the body cells. They are the haploid cells, that is having a single set of the unpaired chromosomes are there. So instead of the 46 chromosomes, we have the 23 pairs. They just have 23 chromosomes in total. 22 autosomes and 1 sex chromosomes or the allosomes. And ovum will always have an X chromosome, but the sperm will have an X or a Y chromosome is there. We already said that the human cell nucleus which contain 46 chromosomes, and when we viewed under the microscope, the 46 chromosomes in a normal somatic cell can be identified by their size, shape and the staining patterns, and the entire set of the chromosomes are arranged in decreasing order of their size. The arrangement is shown in the figure, and in the 22 of the pairs, the homologous chromosomes looks alike and have the same appearance in both the males and females, and these 22 pairs are known as the autosomes, and the two members of the 23rd pair is termed as the sex chromosome or allosomes, and they look different in male and females. In males, the pair consists of two chromosomes are called the X chromosomes, and one X chromosome is also present in the males, but its mate is a smaller chromosomes called the Y chromosomes. The X and Y chromosomes as shown in the image. We can see the sex chromosome X and Y in the image, and when the spermatozoa which undergo meiosis process to reduce its chromosome number, it give rise to two sperms that contain an X chromosome and two sperm that contain Y chromosomes, and the oocyte have no Y chromosomes and they produce only the X containing gametes. If the secondary oocyte is fertilized by an X bearing sperm, the offspring is normally a female, that is XX, and the fertilization by a Y bearing sperm produces a male, that is XY. Thus an individual's sex is determined by the farthest chromosome, that is the SRY chromosome, that is known as the sex determining region of the Y chromosome, which will determine the individual's sex. In the picture we can see the XS zygote and the XY zygote, which is formed by the fertilization process. So up to this we discussed the processes behind the sex determination process. Next we have to discuss how the reproductive system is developed in a human body. So we have to discuss about the development of the reproductive system. The reproductive system developed from the several structures and require chemical substances also. And the gonads, which is developed from the intermediate mesoderm that give rise to the gonadal ridges. And the mesonephric or the Wolffian ducts develop into the male reproductive system, and the paramesonephric or the Mullerian ducts which develop into the female reproductive system. And we will discuss in detail with the help of a diagram. Here we can see the development of the internal reproductive system, and we already said that the gonads, which is developed from the gonadal ridges, and during the 5th week of development the gonadal ridges appears as bulges just medial to the mesonephrons. Mesonephrons are the intermediate kidneys, and adjacent to the genital tubes or the gonadal ridges are the mesonephric ducts are there. It is also known as the Wolffian ducts, and which develop into the structures of the reproductive system in males. And a second pair of ducts, that is the paramesonephric ducts or the Mullerian ducts, which develop lateral to the mesonephric duct, and they form the structure of the reproductive system in the females. And in this picture we can see clearly the mesonephrons are there, and medial to the mesonephrons are the gonadal ridges, and lateral to the gonadal ridges there is the paramesonephric ducts, and the mesonephric duct is there. After the 5th or 6th week, the undifferentiated stages will form into differentiated form, that is male development, and female development will start after the 5th and 6th week. So an early embryo has the potential to follow either a male or the female pattern of development, because it contains both the sets of the duct and the genital ridges that can differentiate into either testes or ovaries. So when we considering the male development process after the 5th week, we already know that the cells of the male embryo have one X chromosome and one Y chromosome is there, and the male pattern of development is initiated by a master switch gene on the Y chromosome, that is named by SRY, and the SRY, it stands for the sex determining region of the Y chromosomes. And when the SRY gene is expressed during the developmental process, its protein product causes the Sertoli cells to begin to differentiate into testes during the 7th week of the development. So this developing Sertoli cells secrete a hormone called the Mullerian inhibiting substance or MIS, and this causes apoptosis of the cells within the paramesonephric ducts, that is apoptosis, that is the death of the cells may causes, and the stimulated by the human chorionic gonadotropin, that is the HCG hormone, the ladying cells in the testes which begin to secrete the androgen testosterone during the 8th week of the developmental stage, and this testosterone then stimulate the development of the mesonephric duct on each side of the epididymis and vas deferens, ejaculatory duct, and seminal vesicles. And the testes connected to the mesonephric ducts through a series of tubules, and these tubules form into the seminiferous tubules, and the prostate and the bulbourethral glands are the endodermal outer growth of the urethra. So this is the male development process happening in the internal reproductive system. And when we considering the female developmental stages, we know that the cells of the female embryo have two X chromosomes only, there is no Y chromosome is there, and because the SRY is absent, the genital ridges will develop into the ovaries, and also the MIS is not produced in female reproductive system, and the paramesonephric duct flourish during the development, and the distal end of the paramesonephric ducts fuse to form the uterus and the vagina, and the unfused proximal portion of the ducts become the uterine tubes or the fallopian tubes, and the greater and the lesser vestibular glands develop from the endodermal outer growth of the vestibules. So this is the developmental stages happening in the internal reproductive system before our birth. So we already said that the Mullerian inhibiting substance, that is the MIS, cause the apoptosis of the cells, that is the death of the cells in the Mullerian ducts in developing males. So no structure developed from the ducts in the male reproductive system. So the testosterone in developing males secreted by the interstitial cells in the testes stimulate the development of the mesonephric ducts into the epididymis, vas deferens, ejaculatory duct, and seminal vesicles on both sides. Here we can see the development of the external genitalis, and external genitalis of both the male and female embryos also remain undifferentiated until about the 8th week. And before differentiation, and embryos have the following external structures, that is the urethral folds, urethral grooves, and genital tubercles, and labioscrotal swellings, and these all are we can see in the images which is given on the left side. And these four structures will form or develop into the external genitalis in the male and female development process. And there is an hormone which is helps to the development or responsible for the differentiation of the external genitalis. And in the male embryos, some of the testosterone is converted to a second androgen called the dihydrotestosterone, that is a DHT. And these DHT stimulate the development of the urethra, prostate, and external genitalis in the male development. And the external genitalis, which may include the scrotum and the penis region, and part of the genital tubercles, which is shown in the figure, it is elongates and develop into a penis region. And fusion of the urethral folds form the spongy urethra and leaves an opening to the exterior only at the distal end of the penis region, and it is known as the external urethral orifice. And the labioscrotal swelling develop into the scrotum. And in the absence of the dihydrotestosterone, the genital tubercle give rise to the clitoris in the female embryos, and the female development will start. And the urethral folds remain open as a labia minora, and the labioscrotal swellings become the labia majora, and the urethral grooves become the vestibules. After birth, the androgen levels declines because the HCG is no longer present to stimulate the secretion of the testosterone hormones. So this is the developmental process happening for the external genitalis. So next we will discuss about the aging and the reproductive systems. During the first decade of the life, the reproductive system is in the juvenile state, and at about age 10, the hormone directed changes starts to occur in both the sexes, and it may lead to puberty. And what is puberty? Puberty is the period when the secondary sexual characteristics begin to develop and the potential for sexual reproduction is reached. And with age, the fertility declines, and when we considering the females between the age of 13 to 14, the ovarian follicles become exhausted and the estrogen levels declined in the female. When we considering the aging and the reproductive system in males, a healthy man often retains the reproductive capacity into their 18s or 19s. At about the age of 55, a decline in the testosterone synthesis may occur, and this may lead to reduced muscle strength, fewer viable sperms, and decreased sexual desires. Although sperm production decreases 50 to 70% between the age of 60 and 80, but abundant sperm may still be present even in the old ages also. And enlargement of the prostate to 2 to 4 times its normal size occurs in most males over the age of 60, and this condition is known as the benign prostatic hyperplasia. So this is the aging and the reproductive system in both the male and female system. So today's section I briefly discussed about the sex determination, development, and aging of the reproductive system. I hope you all understand these three sections. Thank you all for your kind attention.